In this video, I am going to show you how to use manually cropped regions as input to the particle classifier. This feature is useful for manual input because sometimes it is hard to mask the features that we want using automated analysis. So in that case, we have to manually crop the regions of interest that we want to use for our particle classification. So in this case, what we need to do is we can crop an area around the feature of interest. In this case is the protein DNA complex along this piece of DNA. Now there are 3 proteins on this piece of DNA. So we want to draw an inspection box around the feature that we want. To use for our regions of interest. So once we draw this box, we click Save this region of interest. Now we draw one region of interest here, and we now continue to manually draw our second one and our third one. Now you don't have to make the box exactly the same size or shape. It doesn't matter because it's not that important. The particle classifier works by doing a cross correlation between the different particles, right? As long as they are not wildly different, right? Then they should do well in the correlation analysis. Now, in this case, we want, as long as the DNA bending is similar to one another, they should do well in the cross correlation. So that's the idea behind this. Before we go further to doing the correlation, we want to mask the DNA and take note of the mask threshold because we're not using the whole data inside the region of interest as our input to the particle to do the correlation analysis. We can do that, but ideally we just want to use the DNA, but not the background noise, right? So to do that, we can mask the DNA and record that number, which is 2.27 nanometer here. So let me copy that number. Now, after we take note of that number, this is the number we use to mask the DNA so that only the masked area inside this box is used as input to do our correlation analysis. So anything that's outside of the mask inside the box are not used for data analysis. So that's the idea. Then we can go to do particle correlation. Here we have to pick our particle source as the saved region of interest. And the second parameter we need to use is the mask threshold, which is the one that we just write down, right? So it's the same number here. I already did that. So after that, I'm going to click OK, and then it's going to do the correlation analysis using the custom manually drawn regions. Okay, it's finished. Then we can send the 3 regions, right, into the particle classifier. And in this case, we can click region of interest. Now we are inside the particle classifier, and we have the 3 regions or 3 particles here displayed, and you can see that the particles within that region is masked in red, and that's the mask we use to mask DNA, but only the masks within the region are included, right? So outside of that region now, so that way we can localize the feature of interest, which is the protein DNA complex, in this image. Now you see that they are correlated and aligned, and the first 2 particles or regions, they are aligned better as they bend the DNA compared to the third one, which does not bend the DNA that much. So in this case, we can separate these 2 conformations. And if you have a lot of regions, you can further use automated cluster analysis in the particle classifier for classification purpose. And one limitation in this approach is that you cannot see the particle parameters, or rather the region parameters, in the informational panel. It does not work because the regions includes not just one particle, but all the masked features inside that region. So in this case, you see there's a tiny little spot. Let me make that bigger. There is a tiny little spot which is inside the boxed region that we use, but it's not part of the particle. But that's also used for the correlation analysis. So that's one limitation. So it kind of includes some noise around the particle of interest, as long as that noise is masked within our manually cropped region. And because there may be more than one masks within that region, in this case there are 2, so we cannot really display the particle parameters because we don't exactly know which parameters we're looking at, and they cannot be analyzed because these masks are not connected, so they're not a single particle but actually 2 particles, right? So that's the major limitation here, and you can see another spot here. So yeah, so that's the video for this tutorial for using manual cropped region as input to particle classification. Thanks for watching.